Okay, good morning everyone, people in the room and the ones online. Thank you for joining today's seminar. Today we have Nicolas Gompel giving us a talk and I will ask Mathieu Gauthier to introduce him.
Thank you very much, Paul. So it's our pleasure to welcome Nicolas Gompel, who is professor at the University of Bonn and the home of the Haribo candies.
The city of the Haribo candies, yes.
And so he will present us his work on diversification of pigmentation among Drosophila species. So thank you very much. The floor is yours.
Thank you very much, Mathieu, and thanks to the organizers of this seminar series. I hope everybody can hear me okay online and can see me. I'm trying to stay in front of the camera. Yes, so it's very nice to give a talk here, especially because I actually did my PhD in this university in the building that is just next to this one 30 years ago. And I was reflecting that the building has aged better than I did. But in any case, I want to tell you about the work of my lab on the evolution of morphological traits. And we have an issue here, but that I think I need to click this. Yes, and now it should work. Yes. Okay. So generally, I don't think I need to prime this audience much, but the general question I'm interested in, or I've been interested in for now almost 30 years, is to understand how morphological traits or phenotype, other phenotypes such as behavior diversify over evolutionary time. And so, just to illustrate that, I mean, you can capture diversity across the tree of life, or you can focus on very specific groups. And then if you look at live animals, we're not working on these peacock spiders, but this illustrates pretty well the kind of crazy behavior that evolved. But now, what we are interested in is how these traits are generated in the first place. We're not looking so much at how they are being selected, but we're really trying to understand how do they appear in the first place. And so that takes us to looking at the genetic makeup of these traits, or how they form, how they develop. So we're looking at genotypes. We're also looking, in the case of behavior, at how groups of neurons together promote a particular innate behavior. And beyond that, we're of course interested in understanding how they change over time. So my source of inspiration, I'm not going to have more than one slide on details, but my source of inspiration has been to, in parallel to my work as a developmental biologist, I've been collecting insects for nearly almost 40 years now, and I've been looking at, I'm specialized on one particular group. You have one species of that group here, and I've been looking at them. I've been fascinated by their morphology, but the more you look, the more species you look at, the more you see small differences that are used to identify the different species or to tell them apart, and the more you look, the more you see particular patterns of evolutionary changes. And one thing that is striking in this group and in other groups is that they usually share a common body plan, but then on top of this body plan you see small subtle differences. It can be colors, it can be the shape of certain body parts, etc. And as a developmental biologist, I've been wondering, looking at this diversity, where is it coming from? What can we identify different patterns? So this is not unique to this group. If you look at other animals or also in plants, you see this typical thing of variation on a theme. So you have a shared body plan that you can quickly identify, and then sometimes it's pretty crazy. These are tree hoppers, you may be familiar with them, and if you remove the part that is called the helmet that gives all this insane morphological diversity, they all look like cicadas. So all the diversity, or most of the diversity, is coming from this modified organ. And so, when you go to talks on evolution, everybody has their diversity slides, a bit like what I showed, but I was thinking beyond one, spectacular group, are there patterns that we can identify that are shared between groups? And in fact, I think we can. And so I think we can explain a lot of the diversity we see around with a few very simple evolutionary transitions. The first transition is what many of us are interested in, the emergence of novelties. So that is the transition from not having a particular trait to gaining this particular trait that I represent here with a simple circle. But then once you see a new trait appearing in a group, the next thing you know is that it starts to diversify. It becomes a substrate for diversification. So it can be diversity of shapes, it can be diversity of colors or other properties, the expression of some enzymes, etc. And of course, it can be changes in the size of an organ, for instance. So, and I think with that, you explain a lot of the diversity you see in any group of organisms. And of course, there's an additional and perhaps the most common step or transition that we see everywhere, that is going back to square one. So you have a character that has diversified and it's lost secondarily. And so if you think of any group of plants or animals or fungi you're interested in, you think of this transition and you realize that if you have a phylogeny of the group, you can sort of map the characters on top, and these are exactly the transition that explain the diversity you see. So, of course, as experimental biologists, we cannot study the genetic mechanisms underlying this transition in an abstract way. We need a particular group. And for us, we turn to flies, we turn to Drosophila to try to study these different transitions. And our playground has been a bunch of species that are all closely related to Drosophila melanogaster, and that offer exactly what we want. So they are similar enough to facilitate comparison, also at the genetic level, and they are diverse enough, at least in certain traits, to basically study all the transition as I've mentioned. And I'm going to walk you through the different transition I've mentioned. So gain, losses, repeated evolution, I will probably not cover that, but also something that is often not studied enough, I think, by geneticists, which is the quantitative variation. So I keep that for the end. So you see, what you see at a glance here is a lot of variation on the wing pigmentation. There's also variation on the body pigmentation, but we decided to focus on the wings for two reasons. First of all, they offer enough variation, but also the wing has been extensively studied by developmental biologists over the last 4 or 5 decades as a model system to understand development, to understand patterning, and the genetic makeup of the wings. And so, in brief, so what I'm showing you here is, on the left, a gene regulatory network that is active during development, so during the metamorphosis of the animal. And all these genes together set up or organize the primordium of the wing. So their interaction, the details don't matter, but all what you see here will decide where the veins are positioned and where, how much the wing should grow in one direction or the other direction, where are the axes, etc. So all these results is sort of the output of all these interactions that you see here. And these genes are mostly transcription factors, signaling molecules, so they don't really turn the cells into something particular, they just instruct them of their future fate, of their future identity. And in fact, if you look at the expression of these different genes, they paint different territories on the wing. So here you see an adult wing, but in fact, if you go back in time, earlier during development, you find these genes prefiguring the tip of the wing, the posterior compartment of the wing, the future veins, etc. All this is already there at a time when the cells otherwise look the same. And once you have this blueprint, this sort of coordinate system, these very genes, so these are transcription factors, they instruct differentiation genes and they turn each cell into a vein, a sensory organ, a wing blade cell with cuticle, etc., etc. So this is really these two steps where you have the patterning given by these transcription factor genes and so on, and then in turn they instruct the cells to their future fate. And among others, they instruct the formation of pigmentation, which I will be talking about. And so the question, so we had this framework, and we had a tissue that is flat, so it's easy to score phenotypes, to score the pigmentation, and the question we asked is how did the novelty that I showed you on the previous slide integrate in this gene regulatory network? And so in fact, I'm going to start by giving you the answer. So the first transition I will be looking at is the gain of a trait, the novelty. So in our case, it's a simple appearance of a spot of pigmentation at the tip of the fly wings. So this is Drosophila melanogaster, which sort of represent an ancestral phenotype. And here, this is a fly that is called Drosophila biarmipes that has gained this spot, but you can appreciate that they're very similar. The main thing is this spot that is now present on this fly. And so how did this spot appear? So the answer is, in fact, relatively simple. These spots result from the fact that these cells produce more pigment, logically. And why do they produce more pigment? Because all this gene regulatory network is now, has been rewired, and some of the pigmentation genes are now being instructed to produce or to be expressed at higher levels. So basically what I'm saying is that the emergence of a new trait doesn't have to do with new genes appearing. No, it's the same network. It's just the connection between the genes that have changed. And this is what others have found for other traits in other plants, in animals. So this is the main message, I would say, of evolutionary developmental biology. New genes can appear, but most of the diversity we see has to do with the changes in the connection in the networks, in the gene network that control development. And so, so here you would have genes that control pigmentation, and basically we have identified a number of factors that are now directly controlling these pigmentation genes. And so to be a bit more precise, I'm now going to zoom on that. So basically the question becomes, so I'm telling you new regulatory links mean new nodes in the network that didn't exist before. So how does a new node appear? What is a new node in biological terms? So I need to give you a tiny bit of background on how pigments are formed in flies. Basically, this fly has a spot, so this is an adult wing with a spot of pigmentation, and this is because earlier during metamorphosis, there's a number of enzyme, pigmentation genes that have changed their expression pattern. So you find genes such as this one, yellow, that promotes pigmentation, and it's expressed in a pattern that really prefigures the future spot. Here, what you're seeing is it's an antibody staining against the protein yellow, so it's fluorescent. In fact, this wing is completely white. It's completely transparent white. There's no pigmentation yet. But because yellow is expressed at this position, it will later tell the cells that they have to produce pigment at this position. It's not just yellow. There's a number of genes that are expressed either that promote pigmentation or repress pigmentation. And basically, the collective action of all these enzymes result in the simple trait. Right? So we use that to now ask the question at another level, not just asking how the trait evolved, but how these gene expression patterns appear, because it's necessary to have the spot. I guess many of you are familiar with that, but just in terms of background, the way genes are expressed is, so if you have, say, the coding sequence of the gene, you would find a core promoter next to it, and then around the gene coding sequence, you find regulatory element, enhancers. And typically an enhancer will instruct the genes, instruct where the genes should be transcribed and at what time. So that's basically, that would be an enhancer, and it would contact the polymerase that transcribes the genes. And this mechanism will happen only in certain cells. And so here, basically, you have, this enhancer may instruct the gene to be expressed, say, in the abdomen. And so in the abdomen cells, you'll have RNAs being produced, which in turn will be translated into a protein, and then the protein does its job of maybe painting the abdomen. So that's the general framework. How do we find these regulatory elements? You'll see how they are relevant for what we study. If you look at, of course, if you look at genomic sequence, we're pretty good at spotting where the coding sequence is. We can reveal it because we know the genetic code. If you want to identify regulatory elements, it's much more difficult. There's no easy way to predict them, but one way to find them is simply to take sequence. So you say, okay, you have a gene here, and you just assume that there must be regulatory elements around. And so you just take pieces of sequence, and you can then, go to the next slide, you can make what is called a reporter construct. So you take a piece of sequence, you put it in a vector upstream of the coding sequence of a GFP, and then you transform that to make a transgenic animal, and you check in this animal where you see GFP. So if you see GFP in the abdomen, then it tells you that this piece of DNA has all the instructions to determine the expression of the GFP in the abdomen. So you found a regulatory element. So that's generally how people have found a regulatory element. And so in the case of the yellow gene, so I've showed you this slide before, in fact, you can replace this slide by as many regulatory activity driving in the different tissue. So again, this is the yellow gene, you have engraved a coding sequence, and in between, you find all sorts of regulatory element driving expression in the sensory organ, in the bristles, in the wing blade, in the future spot, in the body, etc. There are also elements for the mandibles of the larva and so on. And so here again, each of these elements is active only in a certain cell type. So this one is only active in it triggers the expression of the gene only in sensory organs. So now, so this picture is very typical for what we see for most developmental genes. So you have genes that have a fairly complex expression in different tissues, in different cell types, and the total expression is just the sum of independent activities of this modular element. So that's what has emerged, not just, it's just an example, but this is what we see in general, modular regulatory element that determine the expression in one or the other cell type. So now, with this background, how do we understand the gain? As I told you, it has to do with rewiring of the network, and if we focus on the yellow locus, the difference between the flies that do not have spots and the fly that do have a spot is the expression of yellow is different. So now yellow is expressed in this region, and it was not ancestrally. And why is that? Because there's a new regulatory element that has emerged in the region surrounding the yellow gene. Now, if you align the sequence of a species that doesn't have pigment to the sequence of a species that has pigment, it aligns well. It's not that there's a new piece of DNA that was inserted there. In fact, the sequence has the same size. There are small indels and there are small differences. But what it means is that there are binding sites that have appeared in this region through mutation turnover that have made this sequence capable of driving expression here. And ancestrally, the homologous or the orthologous sequence, I should say, doesn't have this activity. So we wondered what it takes to actually build a new regulatory activity, how many sites, for what factors, etc. So one of the factors we found is this gene distallase. It's one of the transcription factor from this wing regulatory network I showed you before. And now this factor can directly talk to yellow and instruct its expression at the tip of the wing. Ancestrally, distallase also exists. In fact, distallase is a transcription factor that we all express to form our appendages. So all animals express distallase, and that is one of the first steps to specify appendage development. So distallase existed in the wings of other species. It was expressed at the tip of the wing, but it was just not talking to yellow. And now the novelty has to do with the fact that this gene can now, or the product of this gene can bind to yellow and activate its expression. So it seems relatively simple. Now, if I focus on the sequence itself, I mentioned before the fact that we have found a spot element that drives this expression pattern and a wing blade element that drives expression across the wing. And what we see is the sum of both. The expression of yellow is the sum of two independent activities. Now, what we thought was remarkable is that this is an old enhancer. You find it in all Drosophila species, while this one is a recent gain species that have gained this spot. And we thought the yellow locus is about 15 kb. It's quite remarkable that the new element has appeared just next to an existing element already active in the wing. And we thought this may have been a case of co-option where perhaps the wing enhancer environment has facilitated the gain of a new activity in the wing. The problem is that for co-option, you would imagine that it's really the same sequence that has been modified and that the new activity would be sort of embedded in the ancestral activity. And here it doesn't seem to be the case. As I showed you, you can drive the spot with this sequence, you can drive the uniform expression with the other sequence. It doesn't seem to work. But then we thought maybe we're just leaving one parameter out here, which is the amount of expression. Here I'm just showing you a piece of DNA that drives spot pattern or a uniform pattern, but what I'm not telling you is that each piece of DNA is sufficient to drive the full amount of expression that is required. And we thought perhaps to get the endogenous level of transcription, maybe the element or the sequence we need is larger than that. So we revisited that, and I'm going to try and keep it light to ask basically if there's overlap in the instruction for these two activities. So we did a fairly complicated, or we took a complicated detour. So I'm going to try and keep it simple. Basically, we just said, okay, let's pretend that we do not know where these activities are, and let's just map systematically. So we took a fragment that is the entire region, regulatory region upstream of yellow, which we expect to drive all yellow activities in the wings and elsewhere. And then let's just trim this sequence. Let's just trim from the 5' end or from the 3' end to just see how much information we're losing. But then we wanted to not only just say, okay, this sequence has the full pattern. When we cut a bit, we lose part of the pattern. We thought, let's measure. And to measure, we developed a system. Where, so if you look, let's focus on that. I mentioned this reporter assay. So you take a piece of regulatory region, you attach it to a GFP, and you make a transgenic animal, and you take a picture of the wing, and you pick your nicest image, and you say, this is the activity of the enhancer. But then you're biased, because you've picked a nice image. And so what we decided to do is to image for each transgenic line, for each fragment we test, we image 30 wings. And then we can actually deform the images a bit. We use the veins to align all our wings, and we get an average. And so basically the wing you see here is not one wing, it's 30 wings that have been sort of overlapped and aligned, and you get an average which is more representative of the expression. And it's actually, you're thinking, why are you doing that? There's actually a reason. So first of all, we're more objective on the description of the phenotype. We can do that for protein expression, we can do that for pigmentation. Now we can also do the same thing for the abdomen. But there's another reason, that is that we can now all of a sudden take one, okay, I'm going back to my dissection. So we take this big fragment here, and it drives the expression in the spot and some level of expression in the background of the wing. And we say, okay, now if we remove a bit of sequence, what happens? Well, you see that as you trim, you see that the amount of expression goes down. So in the spot, right, it was blue and it fades and then becomes green and then it disappears completely. And same thing for the wing blade, it's pretty bright orange and then it darkens and at some point you have nothing there. And we can actually measure and compare these different constructs, and we can really extract from that the amount of regulatory information that we're losing as we trim. And so that's a way to actually map the information along the regulatory region. So to get to that, so that is here you have the spot activity, and we've determined that all the information for to drive this activity is distributed along this region. It's a complicated mathematical analysis to extract this information. I'm not going to go through that. If you're interested, we can discuss it at the end. But basically what we determine with that is that, you know, these small fragments, maybe 500 base pairs that we thought were the spot enhancer or the wing blade enhancer, it turns out that to get the full level of expression, the region is much larger. Just for the spot, it's basically all it's nearly 4 kilobases of sequence instead of the 500 that we thought were contained all the information. It's just that you add quantitative information. And now with that, we've also mapped the wing blade enhancer, and you see that they occupy the same region. So both are huge, much bigger than what textbooks say about enhancers, and they overlap completely, which is now compatible with the idea that we're looking at a case of co-option. The idea would be that this pre-existing sequence has been used or has facilitated the evolution. We found more to it. We understand more about the mechanism of co-option, but in the interest of time, I'm not going to go into detail. I can just briefly say, to be functional, a regulatory element first needs to be accessible. You know that DNA is wrapped into histones or around histones to form so-called nucleosomes. And when you have nucleosomes, so they are like beads or pearls on a string, and this prevents access from the transcription factor to the DNA sequence. And so the first thing to have an enhancer functional, the first thing that needs to happen is you need to move the nucleosomes away. And there are special instructions in the DNA that gives this accessibility. And the co-option has to do with that. So basically there's some accessibility information in the ancestral enhancer that the spot enhancer has used. And we think that's why it has evolved exactly at this position. But again, we can, if you're interested, I'm happy to discuss that at the end. So long for the gain. And now a pattern that we see a lot, as I said, is secondary loss of a trait. So some years ago, we looked at many species. We have a pretty decent evolutionary or phylogenetic tree for these species. And we mapped the evolution of the spot. So in black, you have all the species that have a spot, and we could infer that the spot was gained once in the ancestor of all these species, and it's been lost at least 5 times independently. And we focused on trying to understand how you lose a spot. And so, for instance, we focused on this pair. I'm not showing them to you, but this one has a really nice dark spot, and this one has completely transparent wings, just like this guy. And they can still mate, actually. You can still get hybrids, but they look very different. And we thought, what did it take to lose the spot? And I'm not going to show you the data. It's an old paper, but basically it's exactly what you would imagine. The enhancer has decayed. So you have the spot enhancer that had been gained here. And then what happened in these species is that mutations have accumulated in the enhancer, and it's still there, but it's just dead, right? So that's sort of an easy way. We found we scrutinized another loss, actually, in Drosophila melanogaster itself. So the fact that this fly doesn't have a spot, it's because it's lost it, or its ancestor have lost it secondarily. And it has lost it in a very weird way. And that's what I'm going to briefly show you now. So if you look at the expression or the activity of this entire upstream regulatory region of yellow, what I showed you before, in Drosophila biarmipes, this is what I've showed you before. In Drosophila melanogaster, uniform expression, so very consistent with the phenotype. But then, for another reason, we precisely look at this accessibility that I mentioned. So we have an assay where we ask where are the nucleosomes present on the DNA, and we looked at both species. So the assumption here is that if a piece of DNA is not an enhancer, you would expect it to be wrapped around nucleosomes. You wouldn't expect it to be accessible. So what you're looking at here, if we look at this line here, for instance, so this is during pupal life. So we've done a development. We look at the halfway through pupal life, and this is the yellow locus, what you're looking at here. And these curves correspond to the degree of accessibility. So whenever you have a peak, it means all the nucleosomes are gone from this region. So what we had expected was that this one is accessible. This one is in red. You can guess where the peaks are behind. And we were expecting it to be accessible because this is where the enhancer is, so very logical. But for melanogaster that has lost the spot, we were expecting the DNA to be completely closed. And instead, what we see here in blue are these two fat peaks of accessibility. And we thought, what? If you lose the enhancer, why should the DNA remain open? You would expect that the signal for accessibility after so many million years would just be lost and that the DNA be wrapped again. And this is not what has happened. So we are very intrigued and we decided to explore what these two peaks were doing. And so the easy way is to, again, take them and make reporter constructs. So now zooming on the non-spotted species of Drosophila melanogaster, again, you're looking at the yellow locus here, and now I'm showing you these two peaks of accessibility. So, right, the peak means no nucleosomes. And so we basically took the region under peak 1, and we made a reporter construct. So hook it up to a GFP, transgenic, what I explained before. And so now, if you take the region encompassing the two peaks, you get the uniform expression very similar to what you get when you take the full fragment. Okay, nothing surprising. Then, when you take peak 1 alone, big surprise, it drives a spot. And we were very surprised because this fly doesn't have a pigmentation spot, and even when you look at yellow expression, you don't see a spot of protein expression. But now, this element taken alone can drive the spot. When we combine it to, so peak 2 alone drives uniform expression, and when you combine them, you could say either this expression is so strong that it masks the spot expression we see here, but we don't think so because we have a sensitive system, and even if there was a little bit more expression, we would probably detect it. Or the sequence here somehow represses what is in there. And in fact, this is exactly what we found. So if we extend, so this is peak 1 that drives this spot. If we extend it a little bit, so we had a bit of sequence, but not enough to get the expression of peak 2. What we see is that now the spot is repressed. So there's, in these 160 something base pairs, there's enough information to actually repress the spot activity that was there and cryptic. So in other words, what I'm saying is that in Drosophila melanogaster, the way the spot has been lost is not by destroying the enhancer. The enhancer is still there. We can reveal its activity, but there's a repressor that has evolved on top of that, and that represses activity. It's completely weird. It's a convoluted evolutionary pathway, and I can skip that. But what we've done is we try to understand how do you evolve a repressor. And it's actually complicated. It takes at least two things. First of all, this region needs to be accessible. And we found that the region, so these 160 something base pair, they have evolved binding site for something that opens up the DNA. We've identified the factor. I don't need to explain how, it doesn't matter. It's a factor called E93, and that's a factor that sort of monitors the progress of metamorphosis. So it's sort of a timer that says, okay, now we need to open this region, and this is during pupil life. But that doesn't create the repression, right? So you first open the region, and on top of that, we've identified, okay, I can skip. This is just to say we found this gene, it binds exactly in this region, and this region is accessible, etc. It doesn't matter. But as I said, you have this new silencer that has been gained, and that represses the spot, and it has gained accessibility. Okay, good. But when you make DNA accessible, you still don't have the repression. The repression is coming from something else. And in fact, we've identified two indels in this region, and if you put the two indels together, like if you remove the sequence that is lost in melanogaster, apparently that has created two binding sites, or potential binding sites for transcription factors, and we don't know what these factors are, but at least one of them mediates the repression. So this site, okay, so here this is the peak 1 plus the extension. So this is the peak 1, the spot plus the repression. And we found two sites in there. If we mutate in particular this one, we see that, we see that the repression is gone, the spot reappears. So it's a bit convoluted, but maybe I can summarize it here. We found that in the ancestral situation, we have an enhancer that drives this spot of expression. The way this spot was lost in melanogaster is by the emergence of a new silencer from the existing sequence, right? It's just binding site being gained through mutations. And so we have a new sequence that can now silence the spot. And what is in this sequence? There's a site that gives accessibility, that moves the nucleosome away, and a site that mediates the repression. So it's like a super complicated way to simply lose a character. And that, I think, reflects very well what François Jacob was describing when he was talking about or comparing the evolutionary process as the work of a tinkerer, just taking bits and pieces and whatever works, but there's not necessarily linear path in the way things evolve. How am I doing on time? Still a bit of time, right? So now if I put all this together, I could say, how does this character diversify? How do you go back and forth between spotted and non-spotted? Well, it's essentially, again, rewiring of these pre-existing networks to gain the spot, and the loss can happen either through decay of an existing enhancer, just mutations accumulating, or in a more complicated way, using this, playing with the accessibility, so making the information available or not. Now I want to move to something that I think has been less explored. So here it was sort of black and white, right? You have a spot, you don't have a spot. But now, if you look at Morphlight, what you see is what you see for other characters. It's quantitative evolution. Just to give a few examples that maybe speak to all of you, the level of curliness or the texture of our hair is evolving or is changing or varying in a very quantitative way. I think you can just look around to appreciate that. This is true for other traits. If you look at these nice butterflies, in terms of patterns, you could say they're quite similar. For instance, they all have a component of pigmentation, of pale pigmentation of their forewings. But then, if you add the color to that, they look very different. And the reason why they look very different is because some properties in these patterns have changed. In that case, the ultrastructure of the surface of the scale has changed, and the way the light is diffracted is different, and that creates different colors. I didn't know that you would attend this seminar when I made this slide, but I think you appreciate that. Same pattern, different properties. That's a nice example. These carpet beetles that you find on every windowsill, different species, they have certain patterns that are more or less similar, but what is interesting is that the same scale or scales at the same position vary in their particular shape. So again, I would say quantitative variation in the form of the scale. So this is a very common way to diversify phenotype. In the case of fly, we can actually study this question at the genetic level because it's not just that flies have a spot or don't have a spot. In fact, the spot varies continuously across species. You see here different species from the group I've showed you before. And so these are actual wings, like one wing from a fly. And here, what you have here is the average over 50 wings from each species. And you see that, our selected wings are actually reflected in the average phenotype. So this is the phenotype per species. Now, if you try to just ignore the pattern for now, and you look at these histograms, so these histograms represent simply the distribution of black pixel on the wing. So intuitively, when you have a lot of pale pixels, you see a lot of pale pixels. You see this peak here, and this shows you that this fly has fewer really dark pixels, right? So it's just how many pixels of each intensity value do I have. And so if you focus on this histogram, you see at a glance that the pale value, the background of the wing, is relatively similar for all species, but the extent of darkness varies. So you have flies that remain relatively pale and others that gain these really dark areas. In fact, if you look at if you just plot the maximum intensity of all these species, you see a near continuum. So the pale value are more or less the same for everyone, but how dark you become it's almost like a dial that you can change. And of course, this is not to insinuate that there's a linear evolution. In fact, you can see among sister species, one may be, I'm trying to pick one that is pretty dark and another one that is much paler. So the spot comes and goes. The intensity comes and goes. So what is determining that. So the answer for us took another detour through this gene yellow, because we know that yellow is necessary to make black pigments. So we thought perhaps the amount of yellow is proportional to the pigmentation. So we tested that. So we took pupil wings, so during metamorphosis, and we did antibody staining. So we revealed the expression of yellow, as I showed you in one of my first slides. So that's what you're looking at here. This is for the Drosophila biarmipes, for the spotted species I mentioned. You see a lot of yellow expression in this region, and the rest is relatively pale. And now, because we have this system where we can overlay the wings at different stages, we can compare the expression of yellow to the adult pigmentation directly. And what we see, so this is the pigmentation, and you see a very good correlation. Basically, wherever you have a dark area, you will have a lot of yellow expressed, right? It's a very good linear correlation. And now we've done that for all the species, or for a number of species we've been studying. And I don't want to go into detail, but every time, so sometimes you have almost a linear correlation, a one-to-one correlation, sometimes not. We know why. There are other genes involved that modulate pigmentation, but by and large, there's a monotonous relationship between the amount of yellow and the amount of pigmentation. So that suggests that the amount of yellow really helps to modulate the level of darkness. So how did that happen? Okay, so this is to show the correlation. So we know where, we know what determines the expression of yellow. We've dissected these regulatory regions upstream, and so we ask whether in these different species that are more or less dark, perhaps these enhancers of yellow have changed their strength, and that may explain the differences we see. And that's exactly what happened. So what we've done for each of these species, so you take a species with a dark spot, we've cloned the regulatory regions upstream, we did a reporter assay, and we measured the amount of expression. So here you have the average expression for these species, and we've done that for all of them, and you can see that the strength, so here you have the pigmentation, and here you have the strength of the enhancer upstream of yellow in each species. And you see that you have species with a very strong, very powerful enhancer, like this one or this one, and you have others that are very pale, and this actually correlates with the pigmentation. So what it means is that we know that there are other genes that also tune the pigmentation. I said that at the beginning of the seminar, but at least this gene yellow, this node, is a diol that helps modulate the pigmentation. It's a regulatory diol. Well, this is just to show that for most species, you have a correlation between the maximum, the strength of the enhancer, if you will, and the amount of pigmentation. But just to, yeah, so now, just to finish, that will be the last part of my talk. I'm saying that somewhere in this region, so we have this 4 kb enhancer, there's information that changes between species that make the enhancer stronger or weaker. And so then the last thing we wanted to know is what does it take to make an enhancer stronger or weaker? Is it just one binding site for distallase that is added and that makes a super strong enhancer, or is the information distributed? So to try and map this information, we compare 2 species that are closely related. That is, you have a good alignment of the sequences, but they differ strongly in their expression. You see this weak expression compared to this darker species with strong expression. And so one is Drosophila biarmipes that I mentioned before, and it's a sister species called Drosophila prostypenis. And this is an alignment. So this is the dark allele, if you will, and the pale allele of yellow here for this species. So again, we are upstream of yellow. And this is the amount of sequence identity. It's pretty good. It's not perfect, but we're probably over 70 or 80% identity. And so that's good, because that means that, yeah, I'm good. We have a good chance to map the genetic changes between these species that explain this difference of expression. And so for that, we decided to make chimeric constructs. So that is, if I take this entire segment, this 4 kb drives that. But now what if I take now a piece of that and I replace the corresponding, the orthologous sequence in biarmipes and I look at the activity. So the idea is, imagine that you have, let's make the hypothesis, you have one site that is a switch to go from pale to really dark. So if you make this chimeric construct, what you would expect is that you take a bit of sequence from the pale species, you replace it in the dark species, there's no change. And you can continue like that until you hit the site that is critical, and then you will switch the activity from high to low. If the information is distributed, meaning imagine it's like many mutations that have appeared along the sequence that explain these differences, then we should find that each piece that we change result in a change of phenotype. That's exactly what we've seen. We've taken this region, we cut it in 6 arbitrary regions, more or less of the same size, and we've done this. So here, just briefly, it looks complicated, it's actually not that complicated. You start from the dark sequence, and then you replace this segment, you put it in the pale sequence. And then here you put 2 dark segments and 4 pale segments, etc. Right? You're just scanning the region one way or the other. And what you see is that as you keep changing sequence, you see a gradual change. You can do it, pale to dark or dark to pale. You see a progressive change of expression, which means, so it's bad news because it means that it's going to be really difficult to pinpoint exactly which site have changed. But it means, and I think it's an interesting message, that this is gradual information. This is a gradual evolution. It's not just one killer mutation that changed the phenotype, but instead it's progressive changes distributed along the entire enhancer that explain the difference. And so, that goes well with the idea that this quantitative change of phenotype is explained by quantitative or by progressive changes in the entire sequence rather than a lucky strong mutation of large effect. And so yes, that's what we add to the scheme I showed you before. In addition to rewiring of the network, I think the quantitative strength of the links is also something that brings diversity. And with that, I have one more thing to say. I'm not going to show you much, but the reason why I'm here is because with Mathieu Gauthier and Arnaud Estouf, we have an INR DFG. DFG is the German funding agency, and we got a grant together after years of fighting to get it. And so the question we want to ask is, what I've told you before until then was what's going on between species, right? But of course, if you look at a population, what we see now is that we've been comparing species with different phenotypes. But the species at some point came from the population of an ancestor. And so we were thinking, what happens at the level of a population? How much variation is there? And if there's variation, how does its structure compares to what we see between species? And so here we can study that, because more than 10 years ago, we collected 500 lines of the spotted species Drosophila biarmipes in India, and now we can do population genetics and combine that to development to ask how the variation within species compares to the variation between species. And we use our system to make them, to put some order. I'm in Germany right now. And then we can measure, we can evaluate the phenotype of each line, and we can compare them, and that's what we've done. I'm going to skip that. It's not necessarily what I want to talk about, but the good thing is that we can now really compare the phenotype within population and between species, and in fact we can compare that in the same morphospace, and we find that there's pretty much as much variation within species in the size and the intensity of the spot as we find between all the species we have. And if we just focus on the intensity, you can appreciate these are different lines from that population, from relatively pale to very dark spot, and this is what we see between species, right? So this is the continuum of intensity within the population, and this is where different species in red would be placed. So it's super because we have comparable phenotype, but at very different time scales. And so that's what we are now set to look at. Okay, so I'm going to skip that, and I just want to finish by acknowledging different people from my lab, current people, past members, different collaborators, of course, including Mathieu and Arnaud. And with that, I'm happy to take a few questions.
Thank you very much, Nicolas. So we can ask questions. Please just only a bit speak up because the microphone is to the front. And the people online can leave their questions on the chat. Yes.
Hi, thank you very much. It was very clear for non-experts in development of biology, like me.
Thanks. Good.
I have a question. What about selection in this? What you observe here actually is already the result of selection.
Mm-hmm.
So you are studying this. How do you think selection has shaped? Was what you observed? And in particular, you presented different mechanisms, some affecting more quantitative versus qualitative, and other qualitative aspects of the patterning. Do you think that some of the mechanisms are more likely to be target selection than others?
So when you say are you talking about what is the selecting agent, or how much selection do we see at the level of sequences?
No, after an initial occurrence mutation,
mm-hmm.
How do you think that, for example, when you present the quantitative, you show that there is a length variation in the length around the enhancer, mm-hmm. Modify the spot intensity. How do you think this has been shaped by selection following in the initial occurrence?
It's hard to know. I don't even know how we would. So maybe I can just say one thing about. So this trait is presumably under sexual selection. The male. So we don't know that from direct experiments, but what we know is that the spot is present in males or in species where the males runs in front of female, display its wings, and whenever the spot has been lost, the behavior has been lost. So we think it goes back and forth. Now, how does that reflect at the level of sequences or in the structure of the enhancer? I don't even know. I think at the level of a population, we can see things, and I think Mathieu has already started to explore that. Between species, it's really hard to say. I mean, we see conserved regions, but we haven't run any analyses that may suggest that a particular region is under selection or not. I mean, I don't know exactly what you have in mind or how you would approach that, but
I think this cut what you are this cross what you showed at the end, so the link between population and species, and so how you can how selection can contribute to go from the population to the species level.
Exactly. So yeah, I don't know. I hope we'll be able to tackle that. The one thing that I can say is that when we did this mapping of, we're trying to see if it was one mutation that explains the variation or if it was actually distributed. The first thing we thought is that enhancers can actually move, right? The sequence may be conserved, but through binding site turnover, an enhancer may move or go in the other direction or expand. And so when we compared these species, the first thing we did was to actually remap the enhancer in the other species, and it has exactly the same structure. So there was no major rearrangement. I don't know what tells us about selection, but at least it tells us that there's no major or fast rearrangement or reorganization of these regulatory regions. I think to start to talk about selection, we would need to be able to focus on specific binding sites where we know that they have a functional effect and see if they are conserved. And we think that's the case for a few of them, but that's all I can say.
Yeah, thanks for the nice talk. I was wondering, in the part where you talk about the loss of the spot, you said that it's kind of a complicated story of the evolution of a new repressor jointly with higher accessibility in this region. So do we know if these accessibility patterns they're genetically determined, or do they kind of follow up by some genes getting more expressed, and it's just kind of
It's encoded in the enhancer. So there's a site. Okay. So there's a gene, I went a bit fast. So there's a gene or transcription factor called E93, and this is one of these factors that can open DNA.
Okay.
And And this factor, in fact, controls the accessibility of thousands of loci during metamorphosis. Some are becoming closed, others are becoming open. And what will make a loci responding to this gene is really binding site in the locus. So what has happened in that case, this new silencer has gained binding site for E93. That's how E93 now binds there and say, open up.
If your idea is that this region became accessible perhaps for other reasons, through this
we don't know.
And then it could allow the evolution of this repression.
In that case, it may have become, so I think the fact that it's accessible is a prerequisite. So if you gain site for something that represses, but the region is not accessible, the site will remain silent. So I think accessibility in that sense is super important in the evolutionary process. But why has it happened like that? I mean, it's, again, we know from other species that you can lose an enhancer with 5 mutations in critical sites. And this is what we've seen in some species. Why would you gain a silencer through, it's a complicated process, we have no idea. Maybe it has to do with, maybe this region does something else, but we don't know. Yeah.
It's related to the previous question about this model that you proposed for the loss of spots. I was wondering, because you found it based on a model with 2 sister species. Right?
Yeah.
So I was wondering whether it was conserved across all the loss of spots. You have elements where it's the same mechanism that get lost.
So we know, we've looked at 2 species that have lost the spot independently, and they use different mechanisms. So one, as I said, is the enhancer. So basically you had this enhancer, you had 3 or 5 mutations, and they kill the enhancer. That's as simple as that. And then we found this other case with this complicated silencing of the enhancer. But in the case of the new silencer, this function doesn't exist in sister species with a spot. Yeah.
So, very much like the talk. At the beginning of the first half of the talk, then you have made a strong assumption of a direct connection between genotype and phenotype, so that one genotype makes this phenotype.
Yeah.
And then in the second one, you have taken a more quantitative approach, and then saying, well, I have made averages. And then the point what I would like to ask is, what is your interpretation of variability, knowing that actually the sites that you're analyzing are very short in very then prone to turnover, then how strong is the impact of transcriptional noise, translational noise, developmental noise? Because one always, from the developmental talks, very often one has the impression that has been a lot of cherry picking for showing the images that we see.
Exactly.
And so what is the actual contribution of noise during gene expression and during development?
Yeah. So we don't know how, and this is something we could address, we don't know how it impacts the phenotype, but we do see, so we make these transgenic lines, right, where we do these reporter assays. So to understand what it means, these lines are derived from one fly. We make a transgene, we integrate it through transgenesis in one fly, and we use this line, we make a line from this fly, and so that means all the individuals we image are supposedly identical, genetically identical. Nevertheless, we observe a lot of noise or a lot of variation among the 50 individuals we image. So why is that? So we know that there's experimental noise. We may stage the flies slightly differently. Among experimentators, we've done that in the lab, different people have tested them, they can get more or less noise, we see that in the PCAs. But it's not just that, there's also individual variation, and we don't know where, I think it's just stochasticity. Mm-hmm. How does that contribute to the phenotype or the phenotypic variation? We don't know, but we could test it. Now we have because we can now look at pigmentation, but we haven't done it. I expect that it's going to be a very loose relationship because these developmental networks are very robust to variation, precisely because there are many, many genes involved. But we haven't, formally in this case, we haven't tested it.
And we have one last question from our guest online, from Nicolas Gompel. I'll read his question.
Yeah.
He says, so the trait is also plastic when the environment changes, like temperature. Mm-hmm. How does genetic variation compares to plastic variation? Is there, I understand, gene-environment interaction meaning that different enhancers respond differently to changes in temperature?
Yeah, I mean, this has been studied, and particularly in the case of this gene and other pigmentation genes that are known to respond to temperature differently. I can say two things. First of all, we do observe variation in pigmentation in response to temperature. If we grow the pigmented fly at 18 degrees or 25 degrees, the spot, the size and the shape of the spot remains the same, the color changes. So it's brown when we grow them at 18 and it's really dark at 25. And this clearly has to do with the amount of expression of the different genes. There's a group in Paris that has studied that, and it has to do with the chromatin structure. So at low temperature, there are some genes that sense the temperature and that will modify histones, and the DNA will be less accessible, and at higher temperature, it will respond differently. So we don't know in the case of the spot what's going on, but generally we know that this is what happens.
Well, thank you very much, Nicolas.
Thank you, everyone.
Thank you.